Hello everyone and welcome to DIPY Workshop 2021. My name is Serge Coudreau and I'm one of the core contributors of DIPY. I'm also the release manager of the project. Today, I will be presenting image registration. Image registration is a quite common step on all processing pipeline. So in reality, you can rarely escape this step if you want to quantify, compare, explain your data results. This is why it is a really important step to understand. This session will be split in 3 parts. So the part 1 is registration basics. We will answer the question why we need the registration step. Answering the why will permit us to understand what is finally image registration. Part 2 is dedicated to image registration methodology and affine registration. From transformation to optimizer and including metrics, we will decompose every step of the registration process and see how we can use it with DIPY. In part 3, we will review some advanced transformation model, and specifically the diffeomorphic model that we have implemented from scratch in DIPY. So why we need image registration in medical imaging? So there is a common issue for dealing with medical images and application. This common issue is a coordinate system. As you can see in this picture here, we have 3 different coordinate systems. The first one is the coordinate X, Y, Z that we call the word coordinate. It refers to usually to the scanner and how the scanner acquires data. The second one is an anatomical coordinate system. So it is represented by the anterior, posterior, the right, the left, and the superior and inferior. This is commonly used in all fields of medical imaging. As you can see in the picture below, neurologists and radiologists have totally different interpretation of this coordinate system. A neurologist will say in this picture that the stroke that you can see in the left temporal lobe caused by the dark area of the MRI data is on the left, compared to the radiologist will say on the coronal view that this same stroke in the dark point is on the right. If you can see also in the axial point of view, the neurologist will say that you see still on the left compared to the radiologist who say that you will still see on the right. So this problem, this interpretation of coordinate system is really problematic. So in a common language, we will say that the radiological works in the last coordinate system compared to the neurologist work in a RAS coordinate system. So the RAS commonly used for left towards the right, posterior towards the anterior, and inferior towards the superior. Then the last coordinate system that is commonly used by all algorithms is a grid system symbolized by IJK. So this coordinate system, the grid space, is our reference coordinate system. This is where we want to register to be exactly. So we will start with the physical space, so the word coordinate, project our data to the grid space in one case, then use the second data, project it to the second part, and you can see that being on this space, we'll have a different scale, size, and orientation that we need to normalize. Also, there is other factor that we need to take in consideration that we want, if we want to normalize our data and compare them. There is, for example, the modality. In medical imaging, we have many different modalities. So we have the CT, the PET, the MRI, cryo, fMRI, OIS. We might want to compare to get more, to combine multiple information. So for this thing, we need registration. We might also want to compare 2D slice with 3D data. We might also want to compare subject in different time points. So it will be, we will always need reference images and the moving images. So this brings us to the question, what is registration? At the end, registration is just an alignment problem. This alignment problem is making a transformation of a set of data into one coordinate system. So the coordinate system that we want is a grid coordinate system. And for performing this spatial normalization, we need usually 3 steps. The transformation model, the similarity matrix, and the optimization method. Concerning the transformation model, we have 2 different types of transformation. We have the classical one, the linear transformation, who apply a global transformation on our image and preserve the length, the angle, and the parallelism. We have the second type of transformation, which is a non-rigid transformation, who apply a local transformation and preserve the diffeomorphism. So here you can see an example in a grid of the different transformation between the linear and the non-rigid. So for the linear, we apply globally in our grid. We can apply some type of rigid transformation, affine transformation, projective transformation. Here's the effect when you apply globally. At the opposite side, you have the non-rigid transformation where you do all this classical transformation locally. If you put this example with brain images, this is what we obtain and what is the difference between a linear transformation and a non-rigid transformation. So you can see that in our case with the brain, all transformation like transformation, rotation, scaling, preserve the different angle and the length and the parallelism of our data. When we apply a nonlinear, we start to deform the space and to deform our data. The second module that we need is a similarity matrix. Metrics are probably the most critical element of the registration problem. The matrix quantifies the similarity between 2 volumes. They measure how well the target object is matched by the reference object after the transform has been applied to it. More the score is higher, more similar are the volume. If we think about it, it is a kind of the inverse of a distance metric. Indeed, in a distance metric, lower are the distance, better is the matching. But how to choose our similarity matrix? It exists a large number of similarity metrics in medical imaging. There is no right image similarity metric, but a set of metrics that are appropriate for a particular application. So here you can see some most common metric that you can find in medical imaging. So the sum square differences, normalized cross-correlation, normalized mutual information, or expectation maximization based. We will go through all this metric in detail in part 2. The third element that we talked before is the optimization method. So the goal of the optimization model is to reach a local optimum for our cost function. In the previous slide, the similarity metric is our cost function. So our goal with picking an optimization method is trying to, with many iterations, to find a local minimum. So that's all for this first overview of the image registration. I hope you like it. Thank you for listening, and I see you on the second part of this video, talking about the methodology of image registration and affine registration. Thank you for listening, and see you on the other part.